Okay, so good morning, everyone, and welcome to the last SIMBA Lunchtime Seminar for 2022. The seminars were planned this year really as a way to introduce people to the SIMBA project and then keep people who already are aware of the SIMBA project up to date on the latest activities. So we've had a series of presenters over the last year, and today we have our last two presenters. So we have Henk Bolhuis from NIOZ in the Netherlands, and then we also have Ching Jian from the University of Helsinki as well. So I think we're going to kick off with Henk. We had a long debate and decided to go in alphabetical order for this. So Henk, I'll hand over to you. And what I'll say is that if anyone has any questions, feel free to put them in the chat, but we will have a brief Q&A at the end as well. So Henk, I'll hand over to you. I think you're muted there.
Typical. Cut it out of the recording, please.
Yeah. Perfect. I'll hand over.
Here we go.
Thank you.
Okay, yeah. So thank you. Indeed, my name is Henk Bolhuis, working for the Royal Netherlands Institute for Sea Research, and also heavily involved in SIMBA. And I will tell you a little bit more about growing potatoes on salt concentrations, and as this jokingly picture a little bit shows you, the potato and the salt is not the best friends unless you want to eat them a little bit salted, but for the growth of the potato plant themselves, it's not the best combination. So a little bit of the background was that especially the coastal regions are very vulnerable to salinification. It's also the place where a lot of agriculture takes place. It's also a place where most of the people live. And especially with the either rising sea levels due to climate change, it's these coastal regions that are endangered by salinification of the soil. It's not necessarily that they immediately flooded with seawater, but it's the salty groundwater level that starts to rise and affects the soils, and so they will become more silty. And this, of course, can be a huge problem because high salt concentrations in terrestrial plants don't mix well together. Now, luckily, we have the SIMBA Horizon 2020 project, which was part of a number of projects that were all more or less on the umbrella of the Coordination and Support Action Microbiome Support. We had Circles, Hollow Food Masters, and SIMBA. And with the SIMBA, we focus on the sustainable innovation of microbiome applications in food systems. And this was the picture of the last meeting in Cadiz, Spain, where we had our meeting. Yeah. I am heading the work package 3 on the marine microbiomes, and it deals with different food types, food sources, and how we can improve them, potentially with microorganisms. This deals with let me see if I can get my pointer back. Yeah. So this deals with seaweeds, with fish farms, with microalgal cultures, but also with the saline agriculture. So these two topics, they should be discussed another time and more extensively, but I'm focusing on the saline agriculture as that was the project that I'm directly involved in myself. So the idea is we have normal potatoes growing on fresh water. They all get very well, very big, huge mass that we create. But if the soil gets more salt, we are going to have a problem. So we will have this increased salinity, and that will have a negative effect on the potatoes, on the plants, and I will show you that later. And so we wanted to study that within SIMBA, and that was, we had, so one step back in my talk. The idea is there are more salt-tolerant potatoes, so you can manipulate the plant genetics or by selective breeding, and so we can get some improve of the potato yields by the selective breeding, but still we don't get back to the normal size and the normal yield that we would like to have. So the idea within SIMBA is, can we use microorganisms or microbiomes, as a collected consortium of microorganisms, to further facilitate this salt tolerance in the different crops. And as I said, I will specifically focus on the potatoes. So on the left side, you see kind of bacterial isolates. Now, a key partner in this was the salt farm on Texel, and they had all these nice test plots with all different, in which they could set the salinity of the different test plots. And so we can do outdoors potato cultivation under perfect conditions like you normally grow your potatoes. And you have your incoming seawater, it's filtered. And so different salinity is perfect condition, perfect. But unfortunately, and this is a newspaper clipping, there was in Dutch, they did not have their financials in good order and they get bankrupt. So this was the basis of my part of the SIMBA research. So it was a huge, huge problem for the whole project. So the biggest problem is, yes, I'm a microbiologist. I'm absolutely not a farmer. Luckily, I have a SIMBA team of people, but, and sorry, you're only here, but they're also not farmers. So we still have the problem. Microbiologists, bioinformaticians, and we want to grow those potatoes. So what we had to do was to find our solutions in-house. And so we allocated the climate room at the NIOZ, and we set up all these racks and tables and boxes and LED panels, and we started to add soils. And we bought the potato tubers for starting the potato plants in the hope that this would work and that this would all go fine. And as you can see, the first attempt was quite successful. So we put in the mud, put in the soil, and then we see slowly the plants growing and even starting to flower, something I didn't know, actually, I didn't expect, but well, it all went fine. So we did these experiments in which we had different salinities. We had the pure microbiome that is already associated with the potato and what is already present in the soil. This was a kind of a blank a baseline measurement. Now, what can be seen is, as expected, a decrease in potato yield with increasing salinity. What we also saw is that the crop actually increased in size, or in weight in this case, with increasing salinity. Can be seen a little bit on this picture. See the plants at the end of the 4 months of growing, they form nice potatoes, but the potatoes themselves, the energy is drained to the potatoes, whereas under high salt we barely find potatoes or no potatoes, and the plant is much more thick and full and heavy in weight. So the microbiome part is we are looking at the microorganisms associated with these different, with the potato roots at different salinities. And we look at the, right, is there somebody talking? Please close your mic.
Okay.
Who is there talking?
Eso parece que, pero bueno, supongo que.
Please, can somebody close their microphone because I'm interrupted.
I think we have it sorted now.
I can continue now. I will continue. So in the rhizosphere, which is the loosely associated bacteria, loosely associated with roots and the directly surrounding soil. And we have the rhizoplane, which are these microorganisms that are more tightly associated with roots, and they're also believed to be have a clear interaction with the plant roots, with the and they may have the most important roles in modulating the plant activity. So we had all these different samples from different salinities, and at a first glance, they all are dominated by proteobacteria, Bacteroidetes, Actinobacteria, and Acidobacteria, or in this case, Verrucomicrobia in the rhizoplane. I'm not going too much into details on the microorganisms. I'm now focusing purely on the salt. What we can see in terms of diversity or in richness, so the number of species and how diverse they are, We actually only see significant differences between the rhizoplane and the rhizosphere communities, but not necessarily between the different salinities they grow at. We also did an NIOZ, a cluster analysis of the different populations. What we do see is a shift in the populations associated with the roots in time. And this is only those 3 samples here, because this is the rhizoplane sample. The rhizoplane, so the roots are only measured at the end of the growth cycle, when we can pull out the plant and the potatoes. Well, these samples are taken from the soil directly from the roots without pulling the plants out, and these are taken at different months during the project. If you look at the different salinities, you can see that there's, well, again, there's the difference between the rhizosphere and the rhizoplane, but there's no significant differences in the effect of the salt concentrations on the microbial community composition. That is the baseline. So still we have the question is, can we improve the growth of these potatoes by doing a transplantation? And the idea is, and I've been working for many years on these kind of salt marshes and microbial mats. These are plants already growing at high salinities. Can we now use the bacteria already associated with these plants that are already adapted to high salinities, to brackish, to marine salt water? Can we transplant that to the potatoes grown under our test conditions and see if we can improve our community? So, and the basis are these coastal microbial mats that just can bind all the sand together, fertilize the sand, the sediment, and then initiate this plant growth. We follow this in time. So we also, again, another baseline was to what happens in nature. So this is the increasing temperatures and increasing, but also the amount of light. This is the summer. Here are the winters. And I measured this abundance in time, and we see also some differences. We see some more shifts at the genus level. We see also some shifts at the phyla level in the rhizosphere of these plants. So there's already a significant variation with time. So what we started doing was isolating bacteria from this salt marsh. Different isolates, characterized them. They were mainly bacillus, some pseudomonas, halomonas, photobacterium, arthrobacter. We also looked at they have plasmids or not. That's for a different study. And then again, we started growing at 0.58% salinity, which was in the middle of this salinity range, where there's still, where they can still increase in growth. The potatoes at too high salinity, it's very tricky to find improvements. Here you have a salinity which is more manageable. And we added the microorganisms and different mixtures. But we also added the natural root microbiome, which was so the natural consortia without first isolating and trying to grow them. We had get some more problems in the climate room, and of course we had COVID, lots of problems, but okay. The results of these experiments, this just finished this summer, was that we found 2 mixtures, a mixture of bacteria mainly consisting of Bacillus rhizobium, some unknown organisms, and Photobacterium. And these improved growth of the salt-grown potatoes. The control is without added microorganism, and we put that at 100%. And we see that we have a further improvement of about 30 to 40%. Still, they don't grow as well as they grow on freshwater, but well, if you have to deal with saltwater, this is the best you can get. Again, we see here in the lower panel that the plants have a kind of an inverse relationship with the potato yield, especially those that grow a little bit less well. Where the potatoes grow less well, the plants grow better. And I think one of the most important take-home messages is that the microbes are, the microbiome is not one thing. It's a variable thing. It depends on the seasons. It depends on temperature. It depends on the amount of the plant growth, the exudates of the plant in the soil, and that's it. So this is a little bit of an overview of what we've done so far in terms of the potatoes grown under laboratory conditions. Thank you.
That's great, Henk. Thank you very much. I don't know if anyone has any questions they'd like to raise your hand, or you'd like to just unmute and ask, you can. Give you a minute, or feel free to add them in the chat. But Henk, I was wondering, so what are the next steps now? So after the growing room, is there plans to take them out into the field then for trials?
Well, that would have been a next step, but that is, I mean, the next step is we have now some leads in microbiomes that may be beneficial. So this mixture number 4 and still the complex salt marsh root microbiome. And so what we would have done in our 3rd year, but due to the 2 years delay, which we're doing in this year, would be to see if we can manipulate some of those microbiomes, pull them apart, make some combinations of the organisms that look promising. And then in the 4th year, which would be 1 year after SIMBA, there we would have been going to the field, but that's unfortunately not possible at all. And yeah, well, that would have been the natural flow. But what I'm now looking for is to see whether we have more molecular identification of the genes of the genomes of these organisms, to see whether we've got specific genes, and so that we can better explain how these organisms benefit these plant growth under salt concentrations. Yeah.
No, it's an interesting. It'll be an interesting next few years, and hopefully, if the research continues on. And I'm wondering, does anyone else have any questions?
Something in the chat, maybe, or is that?
I don't think the.
Oh no, that was Erik.
Oh yeah,
Erik, it was you.
Yeah, I know. I'm sorry. But it looks like, well, just in, it looks, actually, it's a little bit the same as in the ova, right? So it's a very dynamic community. Even, well, in ova, the thing was the same even in constant conditions, it's dynamic. So there's some intrinsic dynamics as well, probably some success.
Yeah, that is a kind of, I think, from SIMBA out, there's no such thing as a microbiome. There are many shifts in the microbiome of a plant or of algae, of the roots. And that, especially when we grow under natural conditions, we have seasons, we have variations in light and temperature and so on. So it is probably very difficult to make one community that will serve all. We may even want to, if you want to do perfect, you may even have to consider whether you should have a spring community and then later on supplement with a summer community. But I mean, these are things for research and to see whether that is indeed necessary or not.
Yeah, that's great. Thank you very much, Henk, and thanks, Erik. So if no one has any other questions, I think we'll move on. Yeah, thanks very much. So I think so what we're going to do now is we'll hand over to Ching from the University of Helsinki. And Ching, if you want to share your screen.
Yes.
Yeah, great. Thank you.
Moment.
Yeah, take your time.
Okay. So yeah, hello. My name is Jin, and I'm working as a postdoc at University of Helsinki, and I have a very different topic. So I will be sharing some preliminary results from a clinical trial that we recently analyzed that's within the framework of SIMBA. So we all know that transitioning to a plant-based diet is good for the environment and maybe health. And we see more and more plant-based products on the market. For example, you see more like plant-based milk, meat alternatives in the supermarket, at least in Europe. But the reality is that the meat consumption is still pretty high in the Western world, and there's also increase in, for example, in China, in developing countries. And the majority of our protein intake nowadays still supplied by mostly by animal proteins, especially in Western world, like you see here, about 60 to 70% of our daily protein intake is actually from animal products. So why is it like people are unwilling to change their diet? I mean, of course, there are some people who are vegan, but for people who have been eating meat or meat lovers, I think it's difficult for them to change their dietary habit if there's no benefit. But people are not so convinced that there will be a benefit by transitioning or by changing their diet to a plant-based diet. And I think the recent study also show that because if you eat more vegetables and plant-based diet, you spend more money because they are expensive, at least in Europe and in the US, and also they are not tasty. So that but some studies show that if they actually know the benefit or health benefit of plant-based diet, they are more willing to compromise on taste and price. And there are some large-scale epidemiological studies that have shown that there's a link of low risk of diseases like cancers and cardiovascular disease and plant-based diet, especially plant proteins. But this kind of observational studies or cross-sectional studies, they are difficult to really pinpoint the mechanism behind the protective effect of plant-based diet. And also it kind of always gives me an impression or give people this impression that, from this kind of cross-sectional studies, you feel like, okay, there's a link between low risk of diseases and plant-based diet, but it's too late for me to make a change anyway. So that we actually need some kind of clinical trial to show people, okay, if you change now, if you change your diet to a plant-based diet now, then you can still get benefit after 3 months. Then that would be more convincing. And also we can actually know some kind of mechanism, because then we can control the exposure. There are some kind of potential mechanism not linked plant-based diet to, for example, low risk of colorectal cancer. But all these kind of mechanisms, they are kind of hypothesized from the evidence in animal studies and in vitro study, which means that the experiment was done in the test tube. So we don't know whether it applies to humans or whether actually when we eat the same meal, the same plant-based meal, we can get the same kind of benefit from what we see in the animal studies. Actually, it's not that easy to actually kind of get the same clinical responses, even if we all eat the same meal, because everyone is so different in terms of physiology and everything. So the big question is that whether there will be any effect or beneficial effect if we actually change our diet from animal-based to a plant-based. So in this study, in this clinical study, we actually try to switch the protein source from animals to plant. And so in one group, actually there were 3 groups, but here we analyzed 2 groups of participants, and they were adult Finnish people, and they are healthy. And one group of people, they were allowed to eat mostly from animal products, and they still eat a little bit, 30% of plant-based protein, but the majority of the protein intake is from animal products. Then another groups, they are like the opposite. They eat mostly plant-based products. And the trial was about 12 weeks. Then we sampled their blood and feces. We did a lot of analysis on biomarkers in, for example, in the serum, and also we did extensive microbiota analysis, because that's like a major component in this whole kind of network linking, diet to diseases. And so, because we generated a large amount of data, so we rely on bioinformatic tools and statistical analysis. And since the analysis is still ongoing, so I just show some kind of preliminary result. So here is the overview of the changes of the the trial. So each circle represents data from one participant. So here we have 46 participants in this animal-based group. Here we have 44. And the red one means there's an increase in some types of biomarkers, and blue means decrease. So you can see that I think the most kind of obvious thing is like there's a very large individual variation, which means that even though some people, they have eaten the same amount of, for example, fat, or here they can be fat or carbohydrate or other kind of macronutrient, they have different responses. For example, in this N-nitroso compound, which is our like non-carcinogen, they can induce colorectal cancer in animals. You can see some spikes in some individuals, and the same in, for example, serum lipids and amino acids. But if you look at the plant-based diet, most participants have decreases values in, for example, serum lipids and amino acids and NOx. And also you can see some differences in terms of the gut microbes. So gut microbiota functions and taxonomy, you can see some differences. And then because we have like so many biomarkers, we try to use this kind of dimensionality reduction method to look at, for example, here one data point is the compositions of serum lipids and amino acid of one participant. So the closer of the data point, the more similar their profiles are. And you can see in this plant-based group, there's a kind of separation of pre-intervention and post-intervention data points, which means there is a difference in terms of their serum lipid profiles and amino acid profiles. Then there's no difference in the animal group. And in terms of bile acids, so bile acids, they are synthesized in the liver and secreted when we eat, for example, high-fat diet, or just in general when we eat fat, because the bile acid will kind of help us digest the fat, so emulsify the fat so that we can digest it. But we know the gut microbes can transform some bile acids into secondary bile acid. Actually, they are the main kind of responsible agent to transform bile acid into secondary bile acids. Then some secondary bile acid can actually induce colorectal cancers in animal models. And here we see in the plant-based diet, there's a decrease in conjugated bile acid and secondary bile acid. Both types of bile acid have been shown in animal studies and in cell studies. They can reduce colorectal cancer or cancer cell growth. Then there is no change in the animal group. Well, if there's any change, there's a slight increase, but it's not significant. Then in terms of NOx and nitroso compounds, we also see like there's a decrease in the plant-based diet, and there's no decrease in the animal-based diet. Then in terms of the gut microbes, so we see there's a slight separation when we look at the overall gut microbiota structure. But because this kind of structure is more like pretty much in a kind of top level kind of difference. So when we look at the compositions of individual bacterial groups, we defined quite many groups that were changed by the plant-based diet. You can see some kind of decrease in many bacteria that are sensitive to bile acid. And also we see this kind of increase in Rosaburia, which is the kind of it degrades fiber. So, you know, it can be because of the higher intake of fiber, so that there's an increase in some kind of this kind of fiber degrader. But there's no difference in the animal-based group. Then we try to associate different biomarkers to gut microbes, and we define the like in the hypothesis, like the gut microbes, they were associated with bile acid and also the circulating amino acid. Then we also found that there is a interesting correlation between one unclassified groups of gut microbes and NOx. So it's a N-nitroso compound. And lastly, we have the kind of interest in identifying who can actually benefit better from switching their diet to a plant-based diet, because we know that different people have different gut microbes, and this kind of microbiota can be actually categorized into different kind of community type, a little bit similar to like blood type. And here we actually can see that in those people who had the more Prevotella before the intervention, they are more responsive to the plant-based diet, but not other types of gut microbiota. So we are still trying to construct a kind of network that link different data set, because we have like so many different data set, like from bile acid, from serum lipids, and so on and so forth. And we try to kind of construct these kind of different data sets and linking this to the gut microbes. And also, I think one interesting kind of thing that other studies before haven't been able to do is to identify what microbes actually can produce NOx or N-nitroso compounds. We know in vitro or in animal studies, gut microbes can form N-nitroso compounds, which are carcinogens. We know proteobacteria like E. coli, they are certainly able to form this, but we don't know who and how they do this in the gut. So we are trying to kind of assemble metagenomes from those people who actually have a spike in NOx after they eat a lot of meat, and we compare to those people who didn't, and we try to see what kind of changes they had in the gut microbes. So this project is part of SkinOppo project, and also within the framework of SIMBA, and the trial was organized by Anna Maria Payari. She is the expert in nutrition, and she also did a lot of experiments in cancer biology, and she also has a lot of other clinical trials on the same topic, plant-based diets and all other kind of plant-based food. And our expertise is in microbiota analysis. We analyze not only the gut microbes, but also other types of microbiota. We also developed our own bioinformatic tool. So if you have any questions or comments or idea for collaboration, you can also contact us. So that's the last. So this is my presentation. Thanks.
That's great. Thank you very much for that. It was just really interesting to hear more about the, I guess, the human side of things. We spend a long time talking about kind of, I guess, field trials and our microbiomes as well. So great to hear the human side. I don't know if anyone has any questions at all. Not yet, anyway. No, I think it was interesting to hear what you were saying about just the kind of the ongoing studies with the gut microbiota. And is that planned within SIMBA as well? Like, will that go on in the next 12 months?
Yeah, because I mean, I'm currently paid by the SIMBA money, so the plan is to use this opportunity to identify relevant NOx producing microbes. That's what I was saying in the last slide, because these NOx producing microbes, they are probably very important if we want to kind of know like who are susceptible to, for example, eating more meat, so that and we try to find this kind of connection, but it is pretty challenging, because we don't know the pathway, for example, in the gut, how this microbe can produce NOx.
Yeah, so it'll be an interesting year ahead. I think, sorry, Henk, you have your hand up there.
Yes. So potatoes are plant-based, but I'm pretty sure you don't want to feed people fish or whatever with only potatoes. So how diverse has the plant-based food need to be? So what would be a kind of a perfect mixture in terms of plant-based foods? Do we need more the leaves, the stems, the roots, etc.?
Yeah, in this study, we focus on plant-based proteins, so that it will be mostly like beans and peas, and also including cereals, and I think also potatoes, including potato, but mostly proteins. And because then, of course, when you talk about plant-based protein, we are talking about the whole food, so that also includes fibers and carbohydrates, so that it's not just one macronutrient, but like mostly like if you eat more plant protein, then overall you eat more like also plant derived carbohydrates and all other stuff in a sense.
Okay, thank you.
I do have another question about just the diet that was prescribed for people. So did you have to give everyone a set menu? Do they have to eat the same things on the same day, or could they just pick from, could they cook for themselves and then keep a diary?
So the trial, the main trial, I had the reference there, but you can also find it from the Anna Maria's publication. But yes, the trial, they supplied I think 90% of their protein source. So that for example, the meat for the meat groups, or the animal, so the plant-based protein like soy or what others, beans, they were supplied by the trial, and the participants were allowed to eat on their own for other types of food. But yeah, the trial supplied most of the main components or the main kind of exposures of this trial. Yes.
Yeah. I was wondering, did you encounter any difficulties with people kind of cheating on their diet and eating things they weren't supposed to?
Yeah, I mean, that is the very common kind of problem with all kind of clinical trials, like people under-report, over-report their diet, for example. But you know, like Finns, they are really interested in joining cohorts. That's why it's good to have the clinical trials in Finland, because they are very health aware. So also we have pretty low dropout rate, so about 5 to 6%, which is lower than usual, like 10-20% that you would expect in normal clinical trials. So that we of course we don't have like very objective method to evaluate their compliance. Not like, for example, in whole grain food, you can actually measure some kind of phenolic compound in the blood to see whether they actually eat whole grain food. But you know, I think the conclusion was in the original publication is that the compliance is pretty good.
Yeah, that's good to know. I was very curious when you were talking about it. It's an interesting one. And I'm wondering, does anyone else have any questions? Feel free to raise your hand, or you can just unmute and ask. No, doesn't look like it. So I think we'll wrap up there. I have to say, Ching and Henk, thank you very much for your presentations this morning. It was great to just find out more about two very different areas of the SIMBA project. So thank you very much. And to everyone who joined us today as well, thank you for coming along, and thank you for attending the seminars all throughout the year. So I think we'll wrap up there. And if anyone has any questions about the presentations today, please feel free to contact me, and I can get more information to you. So that my email address is sarah@aquatt.ie, and you can always find our contact details on the SIMBA project website as well. So thank you very much. I think we'll wrap up there.
Thank you.
Thank
Bye.
you.
Thanks, everyone.
Bye bye.
Thank you.
Thank you. Bye bye.
Bye.
Thanks. Bye.
Bye. Thank you.